Yeah, can you hear me better now? Hello, am I audible? Okay, so let me just start today's session. Good evening, everyone. Welcome to the final extra session for the NPTEL course Animal Physiology, and this is the PMRF-NPTEL session on that. Today, we are going to go through essentially a recap of everything that we have covered throughout the course, and then go through questions and answers which I feel are very important for your upcoming exam as well as overall for the course itself. I will briefly introduce myself. My name is Anubhav Chatterjee. I'm a Prime Minister Research Fellow and PhD scholar at the Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. As you would know that all of these sessions are recorded. All of these sessions are recorded and the recordings are uploaded to this particular YouTube playlist. This playlist link is available on the NPTEL website, and you can also go and search animal physiology on the YouTube and go through them whenever you feel that you need to revise any concepts, or if you have missed the session then you can watch the entire session on YouTube itself. So how we do these sessions is that we are going to do it normally. Okay, I'll just get to that. We will do this normally. I will show you a assignment question from the previous year's run of animal physiology, and this particular week we are going to go through all the 12 weeks and learn about different concepts behind the particular question and understand how exactly to answer them. So I have Lavanya here who is asking, I have forgotten to press the submit button for assignment 12. Missed the submission of assignment 12. Is there any option or excuse? Lavanya, unfortunately I do not think if you have submitted then it counts. Uh, but as you would know that only 8 of the 12 assignments are used to mark the final score. So probably if you have been doing well on the other any 8 of the previous assignments, then I don't think it will be any problem, because this will probably be excluded because you will not be graded for this. Is that fine, Lavanya? I do not think there is any leeway around this, but you can try mailing uh, the NPTEL people and see if they have any solution for this. Okay, we just close the door. Okay, so how we do this is that I will show you this question and ask you to fill in whatever you think is the correct answer out of ABCD into the chat, and then we'll discuss it. So this question is all the way back from week 1 of our animal physiology course. The question is, blank refers to the relatively stable state inside the body of an animal. The options are homeostasis, equilibrium, fluid balance, and organ symmetry. Aruna and Priyanka both say it is A, homeostasis. Surajit and Lavanya also agree that it is A. Raitha and Shantipriya also say that it is A, homeostasis. Jagriti also says that it is A, homeostasis. Okay, anyone else? Okay, so the relative semblance of stability that we have inside our body is known as homeostasis. And homeostasis is maintained by a variety of different mechanisms in our body. The easiest explanation of most of them is there is a stimulus from the external environment. This could be increase in heat, decrease in heat, mechanical stimulus, or auditory stimulus, chemical stimulus, and this affects the homeostasis. Either it increases the level of the stimulus too much, or there is a decrease in the level of stimulus. This particular change is detected by a receptor. The uh, receptor receives the input and passes it along to the control center. Most of the time the receptors are sensory receptors present in our body, mostly on our skin, and the control center here refers to either the spinal cord or the brain itself. The control center then integrates the message and formulates a response, which then it passes as an output to the effector. This effector, as we have seen in the stretch response arc in case, or the reflex action arc that we have also covered, the receptor is one of the sensors present on the skin. The effector here is a muscle which responds in response to the stimulus, and the effector then does its effect, and that essentially brings the body back to homeostasis. And this entire feedback loop continues till homeostasis is reached. So homeostasis is the property of a human biological system where self-regulating process tends to maintain balance for survival. The regulation takes place in a defined internal environment. So here we have different essentially control centers of homeostasis. First we have the hypothalamus present in our brain. This particular guy actually helps maintaining the homeostatic temperature as well as osmotic pressure. We have the skin, which helps regulation of how much temperature there will be in the body via evaporation. Kidneys maintain the water balance as well as the pH balance in our body. Pancreas, as you would know, secretes glucagon and insulin, which help maintain the blood sugar level. The blood throughout our blood vessels as well, especially in the skin, help in dissipation of heat and also help in distribution of heat throughout the body. And finally, in case of severe cold, our muscles start shivering, and this is done by the skeletal muscles, which contract and release heat, which also help in maintenance of temperature homeostasis. Okay, any questions from this? Okay, blank refers to the relative stable state inside the body of an animal. The correct option is homeostasis. Next, the next question is, the appendages that connect muscle to bone are called. The options are ligaments, tendons, cartilage, or nephrons. Priyanka says it is B, tendons. Aruna also says it is B, tendons. Jagruti, Surajit, and Shantipriya also agree that it is B, tendons. Lavanya also says it is B, tendons. Anyone else? Anyone else wants to try this? Yes, go ahead again. Okay, that was something. So yeah, let us. So I have a video for you here which will quickly summarize all the concepts about tendons and ligaments. Share my screen so that you can see it. Okay, just let me know if it is audible or not.
To do what you need to heal, you need to understand the basics of those tendons.
Is this audible? Okay.
Tendon basics. The tendons are connective tissue made up mostly of collagen, and that's somewhat like ligaments and fascia. Now, let's get this out of the way from the beginning so that there's no confusion. Ligaments connect bone to bone, fascia connect muscle to muscle, and tendons connect muscle to bone. You see, when you move your bones, you contract your muscles. But it's not your muscles that pull on that bone, it's your tendons. All the force goes through the tendons, and that means they have to be extremely strong. But tendons aren't just these strong, static tissues, they also have elastic properties. So if you're jumping or running, they work a bit like a spring, storing some of the energy and releasing it on the upward bound. And that's pretty cool. Now let's look at the actual structure of a tendon. Each tendon, like this one, is made up of different bundles. Here's a bundle, there are bundles here, bundles here, bundles here, and finally, here is the collagen fibril. If you have a healthy tendon, it looks a bit like this, with these dense arrays of collagen fibers all parallel to each other. This structure keeps them strong. And opposite of healthy tendons are injured tendons, and tendon injuries are known as tendinopathies. Here's how this works. Tendons are meant to withstand a lot of repetitive loading. But if the load becomes too great, then the tendon gets stressed. They get micro tears. Most of the time your body can repair these pretty easily, yet if the tendon is repeatedly strained so that the rate of degradation exceeds that of regeneration, that is where the problems start. Now, most of these tendon problems happen near the joints, such as the elbow, shoulder, knee, and ankle. Basically areas where we put the most strain on. And if you go to a doctor or health professional, you might hear different terms that describe tendon injuries. The most common term is tendinitis, which describes acute tendon injuries that lead to inflammation. Also, one of the most common things that aging athletes get. That inflamed tendon can hurt. Of course, we understand, right, that inflammation is also the body's natural healing response. Your body's working to heal that tendon, and it's totally normal. But we all want to get past that, right? So how do you heal it? Healing. The first step is to make sure you don't injure it more. That's where some degree of rest comes in. Essentially, change up the activities you're doing to put a bit less stress on that tendon. You don't have to totally stop exercising, and just so you know, the initial repair of that tendon will happen in a few days, but to permanently heal it may take more like 3 to 6 months. You just want to make sure you're not overstraining it again at this stage and permanently damaging the tendon. So you have to be a bit careful, and well, a good diagnosis from a doctor is going to help you know what's up. The next step is rehabbing it. Now, some of the best exercises at this stage are going to be eccentric style exercises. That means exercises where you're letting your muscle lengthen, like lowering a biceps curl. That's an eccentric motion. Lots of doctors now recommend moving, stretching, and gently massaging your tendon while it's healing. The goal of this is to make sure that your healing collagen fibers that make up your tendon end up laying down in the right direction, all parallel to each other, resulting in a strong, healthy tendon. And in the end, just remember that getting the right rehab schedule is really important to healing, and to do that, it's important to get a good diagnosis. If you don't think you're adept enough at diagnosing your own injury, then I highly recommend a good orthopedic doctor.
Okay, that was all about tendons. There's some question here. Okay, so how the less progress, I think you're talking about how the assignments got graded. So how they get graded is that you get graded on the best 8 out of all the 12 assignments that you have submitted, and that will be your grade out of 100. So a 79% is probably like you just take all the marks that you have gotten on the 12 assignments, the best 8 will be used and then extrapolated to out of 100% to give you a percentage. Okay, is that fine, Aruna, or are you talking about something else? There is another thing I think that which is how much of all the videos that you have watched. So if you have watched all of the videos, 100% of them from start to finish, then you will get 100% for the entire course. But if you have watched most of it and some part of it is left for some videos, then you will probably get it lesser. but I do not think that this is used for the final grading, so this is just a statistic that you have with you. Is that okay, Aruna? Okay. So the appendages that connect muscle to bone are called tendons. Any questions from this? What are Merkel cells? So Merkel cells are one of the sensors that are present in the skin, which are mechanosensors, which receive mechanical stimulus such as pain or pinching or any kind of increase or decrease in, you know, the stretching of the muscle or the skin. Is what I remember from them. I will just check that out. Yeah, type of cell in the skin that are involved in touch. So these cells is touch, and then these are the cells which then respond, and these are the receptors that then send the message of touch to the central nervous system where it is identified. Is that okay? Aruna, is that fine? I am not getting clear. Okay, so in your let me show you a picture of it. I think we should be able to get a good image of it. I hope you can see this. Not a very nice picture, but this will make it obvious. Is this visible? So in your skin, in the dermis layer, there are many receptors that are present which sense touch or pressure or heat or any stinging sensation. One of these sensors is known as the Merkel cells. These are these spiky cells that are shown right here, and these cells have a specific morphology, so that when they are pressed or you feel a touch sensation on your skin, they then fire action potential, and these action potentials are detected by the nerves that are associated with these Merkel cells, and they then take the entire information and send it to the central processing unit, so either the spinal cord or the brain, and using these senses you can sense the sensation of touch. So how these have a very complicated but a bit complicated way of functioning, which I do not want to cover in this basic course, but they are actually pretty unique. So if you look at them at Merkel cells or Paschen corpuscles which are present, those have a very unique morphology in how exactly they function. So just to answer your question, Merkel cells are neuroendocrine cells embedded in the dermis layer of the skin, and they are sensors or receptors of the touch on top of the skin. Is that okay, Aruna? Okay. The appendages that connect muscle to bone are called tendons. Moving on. So, actin and myosin are, which are the following: carbohydrates, proteins, fats, and all of the above. Priyanka says it is B. Actin and myosin are proteins. Jagruti and Surajit also say that it is B. Proteins. Lavanya and Shantipriya also say that it is B. Proteins. Preeti also says that actin and myosin both are proteins. Okay, so using this particular question, I would like to discuss about the structure of muscles, which we have covered in some details, but I will discuss it again. So your entire muscles are formed out of very many tubular structures which are present inside them. So if you take a cross section of the muscle, you see this particular unit is made out of smaller tubular structures. This is known as the muscle fascicle. The muscle fascicle, if you then expand it, then you see it is also made out of smaller tubular structures, which are the muscle fibers. And muscle fibers, if you remember how they are formed, muscle fibers are essentially myocytes that are fused together to form long chains. So muscle fiber is essentially a cell which is multinucleated, which is a long lengthened cell which has multiple nuclei embedded in it throughout, and it has a specific uh, stuff inside it which helps in it to actually function as a muscle unit. So what does it have? So if you look even closer, you will see that all the skeletal muscles have this kind of a tubular structure inside it, and this is a striated muscle, which means that you can see bands appearing on the muscle itself. And you would obviously know that the striations are the smallest unit of the muscle, which is known as the sarcomere, as you can see right here. The sarcomere is made out of three important parts. First is our actin filament right here, which is shown in yellow, which traverses the path like horizontally like this, and then in the middle you have the myosin. The myosin is the thicker filament which is present inside the actin filaments and has these protruding parts which are known as the myosin heads. And if you blow up the actin looks like this. This is the yellow trail of actin, which is a filament itself, and on top of it you have two more proteins which follow it, which are tropomyosin and troponin, and this entire unit is known as the sarcomere. In the middle of the unit, where you have mostly the myosin, is known as the M line. Then you have the Z discs at the end, and the myosin is attached to the Z disc via titin. So this is the entire sarcomeric unit. So here we have a video to show you exactly how myosin and actin work. Just let me know if this is audible.
What I want to do in this video is try to
Is this audible? Okay.
understand how two proteins can interact with each other in conjunction with ATP to actually produce mechanical motion. And the reason why I want to do this, one, it occurs outside of muscle cells as well, but this is really going to be the first video on really how muscles work, and then we'll talk about how nerves actually stimulate muscles to work. So it'll all build up from this video. So what I've done here is I've copied and pasted two images of proteins from Wikipedia. This is myosin. It's actually myosin 2 because you actually have two strands of the myosin protein. They're interwound around each other. So you can see it's this very complex looking protein or enzyme, however you want to talk about it. And I'll tell you why it's called an enzyme, because it actually helps react ATP into ADP and phosphate groups. So that's why it's called an ATPase, or it's a subclass of the ATPase enzymes. This right here is actin. This is actin. What we're going to see in this video is how myosin essentially uses the ATP to essentially crawl along. You can almost view it as an actin rope. And that's what creates mechanical energy. So let me draw it. I'll actually draw it on this actin right here. So let's say we have a myosin, one of these myosin heads. So when I say a myosin head, this is one of the myosin heads right here. And then it's connected, it's intertwined, and it's wove around. This is the other one, and it winds around that way. Now let's just say we're just dealing with one of the myosin heads. Let's say it's in this position. Let me see how well I can draw it. Let's say it starts off in a position that looks like that. And then this is its This is kind of the tail part that connects to some other structure. And we'll talk about that in more detail. But this is my myosin head right there when in its starting position, not doing anything. Now, ATP can come along and bond to this myosin head, this enzyme, this protein, this ATPase enzyme. So let me draw some ATP. So let's say so ATP comes along and bonds to this guy right here. So let's say and it's not going to be this big relative to the protein, but this is just to give you the idea. So as soon as the ATP binds to its appropriate site on this enzyme or protein, the enzyme, it detaches from the actin. So let me write this down. So 1, ATP binds to myosin head. Myosin head. And as soon as that happens, that causes the myosin to release. Myosin releases actin. So that's step 1. So I started off with this guy just touching the actin. The ATP comes and it gets released. So in the next step, so after that step, it's going to look something like this. And I want to draw it kind of in the same place. So after the next step, it's going to look something like this. It will have released. It will have released. So now let me shade it in with the white. So now it looks something like that. And you have the ATP attached to it still. I know it might be a little bit convoluted when I keep writing over the same thing, but you have the ATP attached to it. Now, in the next step, the ATP hydrolyzes. The phosphate gets pulled off of it. This is an ATPase enzyme. That's what it does. So let me write that down. So step 2, let me scroll down a little bit. Step 2, ATP goes to ADP plus a phosphate group. And what that does, that releases the energy to cock this myosin protein into kind of a high energy state. So let me do step 2. So step 2, this thing, you can kind of view, it gets hydrolyzed, it releases energy. We know that ATP is the energy currency of biological systems. So let me draw it. So it releases energy. I'm drawing it as like a little spark or explosion, but you can really imagine it as it's changing the conformation of, it kind of spring loads this protein right here to go into a state so it's ready to kind of crawl along the myosin. So in step 2, so plus energy, and then this, you can kind of say it cocks the myosin protein or enzyme to high energy. You can kind of imagine it kind of winds the spring or loads the spring to high energy conformation. And conformation for proteins just means shape. Conformation. So the step 2, what happens is the phosphate group they're still attached, but it gets detached from the rest of the ATP, so that becomes ADP. And then that energy changes the conformation so that this protein now goes into a position that looks like this. And let me draw it like, so this is where we end up at the end of step 2. Let me make sure I do it right. So at the end of step 2, it might look something like this. It looks something like this. I'm trying my best to draw it. So at the end of step 2, the protein looks something like this. This is kind of in its cocked position. It has a lot of energy right now. It's kind of wound up in this position. You still have your ADP. You still have that's your adenosine. And then let's say you have your 2 phosphate groups on the ADP. And you still have 1 phosphate group right there. Now, when that phosphate group releases, so then let me write this as step 3. So remember, when we started, we were just sitting here. At the ATP kind of binds on step 1. Actually, it does definitely bind. At the end of step 1, that causes the myosin protein to get released. Then, after step 1, we naturally have step 2. The ATP hydrolyzes into ADP phosphate. That releases energy. And that allows the myosin protein to get cocked into this high energy position and kind of attach, you can kind of think of it, to the next rung of our actin filament. Now we're ready to we're in a high energy state, let me write this down. In step 3, the phosphate released from myosin. The phosphate is released from myosin in step 3. That's step 3 right there. That's a phosphate group being released. And what this does is this releases that energy of that cocked position, and it causes this myosin protein to push on the actin. This is the power stroke. If you imagine in an engine, this is what's causing the mechanical movement. So when the phosphate group is actually released, remember the original release is when you take ATP to ADP in a phosphate. That put it in this kind of spring-loaded position. When the phosphate releases it, this releases the spring. Releases the spring. Releases the spring. And what that does is it pushes on the actin filament. It pushes on actin filament, pushes on actin. So you could view this as the power stroke. We're actually creating mechanical energy. So depending on which one you want to view as fixed, if you view the actin as fixed, whatever myosin is attached to would move to the left. If you imagine the myosin being fixed, then whatever the actin and whatever it's attached to would move to the right. Either way. But this is where we fundamentally get the muscle action. And then step 4, you have the ADP released. ADP released. And then we're exactly where we were. We're exactly where we were before we did step 1, except we're just one rung further to the left on the actin molecule. So to me, this is pretty amazing. We actually are seeing how ATP energy can be used to, we're going from chemical energy. Let me write that down. We're going from chemical energy or bond energy in ATP, chemical energy, to mechanical energy. Mechanical energy. And for me, that's amazing, because when I first learned about ATP, and people say, oh, they use ATP to do everything in your cells and contract muscles, I thought, well, gee, how do you go from bond energy to actually contracting things, to actually doing, what we see in our everyday world as mechanical energy? And this is really where it all occurs. This is really the core issue that's going on here. And you have to say, well, gee, how does this thing change shape and all of that? And you have to remember, these proteins, based on what's bonded to it and what's not bonded to it, they change shape. And some of those shapes take more energy to attain. And then if you kind of do the right things, that energy can be released, and then it can push another protein. But I find this just fascinating. And now we can build up from this actin and myosin interactions to understand how muscles actually work.
Okay, so that explains pretty much the basics of how actin and myosin work to essentially provide the movement of the entire muscle. I will be going through it in a bit more detail. Yeah. So, as you just saw, I will just start here, back here. So this is the sarcomere, in which the actin and myosin are arranged like this, with the myosin being in the middle with its heads pointing outwards, being attached to the actin, which is on both sides of it. So how does exactly the entire
This video. So what I've done
thing function? So here, if you see a zoomed in picture, this is the myosin, which all of its heads pointing outwards like this. And here's the actin filament. So in order to work, the myosin has to come in contact with the actin on the active sites present on the actin. But these are all blocked by the tropomyosin molecule. This is also a filamentous molecule that goes and wraps all the way around the actin and blocks all the active spots. So in order to work, you have to somehow remove this particular molecule, tropomyosin, out of the active spots. This is done in the presence of calcium. calcium binds to the third molecule present here, which is troponin. And troponin, once bound to calcium, removes tropomyosin from the active sites, allowing the myosin heads to bind to the active filament, actin filament, and produce the mechanical movement. So how exactly what exactly actin and myosin are? The correct option is protein. Next we will discuss about the mechanism of actin and myosin movement. Any questions from this initially? Does anyone have any questions from this? I guess not. So moving forward, blank leads to actin myosin binding and blank leads to their detachment. The options are ATP leads to actin myosin binding and blank leads to their detachment. ATP ADP, calcium ions and ATP, calcium ions and ADP. We have a question here, which is a bit separate from this. Just while we are getting the answer, I will see this. How a diastolic and systolic is calculated in BP. So whenever you diastolic and systolic are two types of pressure that you can actually measure when you are using a sphygmomanometer or the blood pressure measuring machine. So both of them actually correspond to one, the highest pressure that you feel in your blood vessels, which is when the left ventricle has just pumped the blood out, and while it is going through your arteries at full force, that is the highest pressure that you can measure. So that is your diastolic pressure. And once in between or left ventricle pumping, after a while the blood starts flowing back, so the arteries have dropped down to a lower pressure when there is no force pushing the blood through them actively. That is the systolic pressure. So these are the both of them. Actually it is opposite, systolic and diastolic. Systolic is the higher pressure that you can measure in the blood, and diastolic is the lowest most pressure that you can measure in the blood. Both of them are measured using the sphygmomanometer, which gives you an indication which pressure you are measuring. Okay, so coming back to the question, we have Lavanya answering it is C. Calcium ions lead to binding of actin and myosin, and ATP leads to their detachment. Surajit and Jagrati also say that calcium ions lead to binding of actin and myosin, and ATP leads to their detachment. Anyone else? Okay, so let us look at it closely. So just as we have just discussed, so this is a blown up picture of a myosin filament, and you can see all the heads that are pointing out of it, followed by a actin complex. Actin complex, if you zoom in here, as I said, consists of three different parts. The green actin that is going all along, then you have tropomyosin, which follows the active sites of the actin and blocks all of them, and troponin, which binds to tropomyosin and actin. And here you have the myosin molecule, which has, so these pointy ends are the heads of the myosin molecules. Here is the tail, which attaches to the overall filament, and the head right here, which has actin binding sites as well as ATP binding sites. So this is a bit busy diagram, but I would like you to follow along. So this is the resting phase, the topmost, the north side phase. So actin is present here, myosin is present here, tropomyosin is blocking the active sites, and myosin in this condition is bound to ADP and inorganic phosphate. It has already used up a ATP molecule to which it was initially bound. So next, calcium binds to troponin right here, and calcium binding to troponin removes the tropomyosin from the active sites of the actin, allowing the myosin to go and bind to the active sites. Once this binding occurs, then comes the power stroke, which is the movement of the head of the myosin. As you can see right here, it moved in this particular direction. So either the entire myosin contracts, and this actually provides the contraction of the entire muscle. So this is the smallest unit of contraction that actually contributes to the entire contraction of the muscle. Once this occurs, another ATP goes and binds to the myosin head, which brings it back to its cocked position. So it goes back to its initial position, and this entire process is repeated again and again. Here is another diagram which shows the same. When there is calcium, then the ADP bound myosin head binds to the actin. ADP bound myosin head is charged and performs the power stroke and pushes the actin in one direction. This bending actually causes release of ADP, and finally a new ATP molecule binds, which causes the ATP to be utilized and return back to the cogged stage of the myosin head, and this entire process repeats. So if we look closely, the binding of ATP actually dissociates actin and myosin, and the binding of calcium actually brings actin and myosin together. So blank leads to actin myosin binding, and blank leads to the detachment. Calcium ion leads to the binding of actin and myosin, and ATP leads to its detachment. Any questions from this? Okay, moving on, we go into talking about the neural systems. The first question from that is, resting membrane potential of a neuron is blank, and the negative sign indicates that there are blank outside of the cell than those inside the cell. So the options are A, resting membrane potential is minus 70, and there are more positive ions outside the cell than inside. Then we have the opposite, the resting membrane potential is minus 70, but there are more negative ions than outside. Then resting membrane potential is minus 10, and there are more positive ions outside than inside. And finally, resting membrane potential is minus 10, and there are more negative ions outside than inside. So yeah, so we have the first answer coming in. Surajit says it is A, which is minus 70 is the resting membrane potential, and there are more positive ions outside than inside. Lavanya says it is minus 70, but there are more negative ions outside than inside. And Jagruti also says A, minus 70 and positive ions. Anyone else? Anyone else want to try and answer this? Okay. So here again I have a video for you to see. Just let me know if this is audible or not.
In this video, I want to talk about how the neuron...
Is this audible?
resting potential is created and how it relates to concentration differences in some of the important ions involved in neuron function, because understanding the concepts involved in the neuron resting potential will help us understand other neuron membrane potential changes like the graded potentials and the action potentials. So here I've drawn kind of a blown up neuron. So we'll have the soma in red, the axon in green, and one dendrite in blue, and I've blown up the dendrite and the axon really large just so I have some space to draw. It's useful to consider the formation of the neuron resting potential in imaginary steps, but in reality they all occur simultaneously. So first, let's consider a neuron with no resting potential, so it's not more positive outside or more negative inside the membrane, and we'll consider that all these key ions have the same concentration inside and outside the neuron, so that there are no concentration gradients for organic anions, potassium, sodium, chloride, or calcium. Now the neuron is going to be creating organic anions and adding them to the cytoplasm. And as organic anions are being created, most of which are proteins with a net negative charge, these extra negative charges that are being added to the inside of the membrane are going to create a small membrane potential, so that now it'll be more negative inside the membrane of the neuron because of these extra negative charges. But this will be something small, like maybe it'll be somewhere around -5 millivolts. I don't actually know what this would be, but it's going to be some small membrane potential that's not going to be enough for the neuron to function. But even with a small membrane potential, there will now be an electrical force acting on the organic anions, because they will be attracted to the more positively charged outside of the membrane, so that the electrical force will be trying to drive organic anions out of the neuron, but they won't be able to leave, because it turns out that the membrane is highly impermeable to the organic anions. And the same will be true for the diffusion force caused by the higher concentration of organic anions inside the neuron compared to outside the neuron. Even though these electrochemical driving forces are trying to make the organic anions leave, they can't get past the neuron membrane, so they're trapped inside the neuron. So no changes will happen to the concentrations of the organic anions, and no further changes will happen to the membrane potential at this imaginary step. The same is not going to be true for the other ions, because the neuron membrane has channels in its membrane called leak channel or leakage channels. And these channels allow these ions to pass across the membrane, although how easily these ions can cross the membrane through the leak channels is very different for the different ions. And these leak channels are open all the time. They're not gated, so they're not opening and closing in response to some kind of stimulus. For the next imaginary step, let's consider what happens when we add the sodium-potassium pump to the neuron membrane. So this is going to be our sodium-potassium pump. This will be an active ion transporter. It will use the energy of one adenosine triphosphate molecule, that I'll write as ATP for short, to actively transport 3 sodium ions outside the neuron, in exchange for 2 potassium ions being transported to the inside of the neuron. Now, by moving more positive charges outside the neuron than it's moving inside the neuron, that's going to make the membrane potential bigger. It's going to make it more negative inside the neuron membrane. I'm not sure exactly what size of change that would be, but it's going to be something small. Let's just say, for example, it might be around -10 millivolts. So now we have a bigger membrane potential, but it's still probably too small for the neuron to function. Far more importantly than this change to the membrane potential, however, is that we're going to have a big change to the concentrations of potassium and sodium inside the neuron. By pumping potassium inside the neuron, we will now get a much larger concentration of potassium on the inside of the neuron than we have on the outside of the neuron. And by pumping sodium outside of the neuron, we're now going to have a much smaller concentration of sodium on the inside of the neuron compared to the concentration of sodium on the outside of the neuron. Now you may be wondering why the concentrations of potassium and sodium changed inside the neuron, but they didn't change on the outside of the neuron. And the reason for that is that the extracellular fluid all of this fluid outside of the neuron is huge, and the total number of ions outside the neuron in the extracellular fluid is very large compared to the volume of the cytoplasm inside the neuron and the total number of ions inside the neuron. So that any of these movements of ions across the membrane of a neuron is really going to only change the concentration of ions inside the neuron, and any change to the concentrations of ions in the extracellular fluid is going to be negligible. It's going to be so small it's not even worth considering. There are now opposing forces on the potassium ions. The electrical force will be trying to drive these cations in to the more negative inside of the cell, but the diffusion force will be trying to drive potassium out of the cell from a higher concentration to a lower concentration. At typical neuron ion concentrations at this imaginary step, the diffusion force would actually be much larger than the electrical force. So while there are forces pointed in opposite directions acting on potassium, the net electrochemical driving force, that is the larger diffusion force minus the smaller electrical force, will cause a net movement of potassium ions out of the neuron through the leak channels. As potassium ions are leaving the neuron and carrying their positive charges out of the neuron, that is going to make the inside of the neuron membrane more negative. The membrane potential is going to get bigger and bigger with each potassium ion that leaves. And it's going to continue to do so until the membrane potential is big enough that the electrical force trying to drive potassium ions into the neuron is the same size but in the opposite direction as the diffusion force trying to drive potassium ions out of the neuron. When these forces are the same size and pointing in opposite directions, there is no net movement of potassium ions into or out of the neuron. The occasional potassium ion will wander out or wander into the neuron, but in basically even amounts. For potassium, at typical neuron ion concentrations, this might occur around negative 70 millivolts, which is more than enough of a membrane potential for the neuron to function. The membrane potential, where an ion has balanced electrical and diffusion forces so that there's no net movement of that ion across the membrane anymore, is called the equilibrium potential. It's also called the reversal potential. Now, it actually doesn't take that many potassium ions to exit the neuron to reach the equilibrium potential for potassium. It's something like less than 1% of all the potassium ions in the neuron have to leave for potassium to reach its equilibrium potential, so that the effect on the intracellular concentration of potassium is really negligible. This process does take a little time, though, because most of the membrane is impermeable to potassium, and it can only squeeze out through the leak channels. Now, for sodium ions, both the electrical force and the diffusion force are strongly trying to drive sodium into the neuron because it's a cation, so it's attracted to the negative inside of the neuron membrane, and its concentration is much higher outside the neuron than inside the neuron. If we had a membrane that was only permeable to sodium, these cations would flow into the neuron until so many positive charges entered the neuron that it actually wouldn't be negative inside anymore. It would actually become positive, at which point the electrical force would switch, and it would have to get quite positive inside the neuron for the electrical force to get big enough to balance the diffusion force to reach the equilibrium potential of sodium, which at typical neuron ion concentrations may be around positive 50 millivolts. Without input, however, when the neuron membrane is at rest, the permeability of the membrane to sodium is actually quite a bit less than it is for potassium. And in fact, the permeability of the resting neuron membrane to sodium is something like 4% of that of potassium, so quite a bit less. But because some sodium is able to squeeze through these leak channels, bringing some positive charges into the neuron, that is going to affect the membrane potential a little bit, so that instead of being at the potassium equilibrium potential of around negative 70 millivolts, at this imaginary step it might settle in around negative 60 millivolts. And that's actually a pretty typical neuron resting potential. When the membrane is permeable to multiple ions that have electrochemical driving forces, the resulting membrane potential is a weighted average of the equilibrium potentials of those ions, weighted by their permeability. At rest, the neuron membrane is much more permeable to potassium ions than it is to sodium ions, so that the resting potential is much closer to the equilibrium potential of potassium than it is to that of sodium. Because the concentrations and permeabilities of ions are usually stable in most resting neurons, the resting potential is usually stable as well. Now neither potassium nor sodium is at its equilibrium potential, however, so there will be a small amount of net movement of both ions across the membrane. A little bit of potassium will be dribbling out of the membrane, and a little bit of sodium will be dribbling into the membrane. This will be matched by ongoing activity of the sodium-potassium pump, which not only creates these concentration gradients in the first place, but also maintains them over time. The resting membrane usually has an intermediate permeability to chloride ions of around 45% of the permeability to potassium ions. In contrast to potassium and sodium, whose concentration gradients play a big role in creating the resting membrane potential, for chloride, the resting membrane potential plays a big role in determining its concentration gradient. The membrane potential drives chloride out of the neuron until its concentration gradient is big enough to balance it, so that normally there's a very small concentration of chloride ions inside the neuron compared to the outside. And because of that, the equilibrium potential for chloride is usually at or close to the typical resting potential for a neuron of around -60 millivolts. Most neurons have other active means of decreasing the intracellular concentration of chloride, though, the main one being the chloride potassium symporter. And this drives chloride out of the neuron by harnessing the diffusion force acting on potassium ions. So it lets potassium move out of the neuron like it wants to, and it harnesses that energy to also push more chloride ions out of the neuron, further decreasing the intracellular concentration of chloride. Because of this, the equilibrium potential of chloride for most neurons is usually not at the resting potential of around -60 millivolts, but instead it's around -70 millivolts, which will cause some inward chloride flow, leading to a minor change in the resting potential to a more negative value, although usually the change is negligible. Most neurons also have active means to decrease the intracellular concentration of calcium, so that there is a small concentration of calcium ions inside the neuron compared to the outside of the neuron. The main one of these being the sodium-calcium exchanger, which I'll show right here, that drives calcium out of the neuron by harnessing the energy of the electrical and diffusion forces acting on sodium, so that the calcium-sodium exchanger allows sodium to enter the neuron in exchange for pumping calcium out of the neuron. And this creates strong electrical and diffusion forces trying to drive calcium into the neuron. And these forces are so big that the equilibrium potential of calcium wouldn't be reached until the inside of the membrane was around +120 millivolts to flip the electrical force to balance the diffusion force trying to drive calcium into the neuron. But it turns out that the resting membrane permeability to calcium is actually quite small, so that it usually has little effect on the resting membrane potential. So while the concentration gradients of chloride and calcium ions tend to have little effect on the resting membrane potential of most neurons, they do play major roles in other aspects of neuron functions, which we'll talk about in later videos.
Okay, so that should nicely summarize how exactly the membrane potential is found in neurons. I'll just introduce you to one other concept. So it was all well and good that we had an experimental basis for the entire problem of how exactly membrane potential works, but we did not essentially have a theoretical basis of how exactly to calculate the membrane potential. This was given by the Goldman-Hodgkin-Karts equation, in which the equation is this guy right here, which is EM, which is the membrane potential, equal to RT by F log of permeability of a particular ion multiplied by the concentration of the ion outside divided by the permeability of the ion multiplied by the concentration of the ion inside. And this, if you see, actually uh, are similar to the concepts that we have already seen in the video. The permeability, as we have seen, depends on the ion channels. The concentration difference depends on how exactly the membrane is set up. And thus this particular equation nicely summarizes the entire experimental problems. And with only this much knowledge of the permeability of the ions and the concentration of the ions, you can actually figure out the membrane potential. Experimentally also, membrane potential can be determined, and it is determined using a very simple voltmeter arrangement, in which you have a particular, so the voltmeter is a device which can detect voltage, and the ones we use to detect membrane potentials are very sensitive and have two ends, one of which are probed inside the neuron being questioned and one kept outside in the medium. And the voltmeter will give you the difference between the potential on the outside and the inside, which will be your membrane potential. So coming back to the question, the resting membrane potential of a neuron is blank, and the negative sign indicates there are more blank outside than the inside. The correct option is 1A, which is it is minus 70, which is the resting membrane potential, and there are more positive ions outside than inside, and that is why the resting membrane potential is minus 70. So we have had a question here from Aruna. Kindly explain the respiratory mechanism, nervous and chemical control. Okay, so I don't think this was covered in the lectures. So the nervous control of the respiratory mechanism includes two types of nervous systems, which is the parasympathetic and the sympathetic system. The sympathetic system is our fight response, and the parasympathetic is the sympathetic is the fight or flight response, and the parasympathetic is the rest and digest response. So under the stimulation of the sympathetic nervous system, which stimulates, essentially innervates the entirety of the lung starts exchange of oxygen to the underlying blood much faster, and the opposite happens in the case of stimulation by the parasympathetic system. And the other one, the chemical control of respiratory mechanism, is done via various uh, chemisensors in our body, and these guys are present both in our neck as well as close to the hypothalamus, and these detect the partial pressures of oxygen as well as carbon dioxide present in our blood, and depending if there is an imbalance between the levels of oxygen and carbon dioxide, this information is transferred to the hypothalamus, which then uses its nervous control to maintain and increase the rate of respiration via the lungs. So you can actually go through it in a bit more detail. I think there are lectures on YouTube that will help you go through it, and there is the book by Arthur C. Guyton, I think, Medical Physiology, which covers this topic very nicely. Is that okay, Aruna? Okay. Moving on, a redistribution in electrical charge of a cell occurs. It leads to less negative charge inside the cell than outside. It is called hyperpolarization, polar peaking, depolarization, or sinoatrial faulting. So there is less negative outside than inside the cell, so it is called hyperpolarization, polar peaking, depolarization, or sinoatrial faulting. You can put whatever you think is the correct answer into the chat. Surajit says it is A, hyperpolarization, when it is less negative inside the cell than outside. Aruna says it is C, depolarization, which is less negative charge inside the cell than outside. Jagruti also says it is C, depolarization, when it is less negative charge inside the cell than outside. So I had a video on neuron action potential, but I don't think I will just go through the slides here. So we have already learned how exactly the membrane potential of a neuron or any cell is set up via the various ion channels that are present on the surface of the particular cell. So we will focus our discussion on three important types. First is the sodium potassium transporter. This particular channel actually allows the uh, export of sodium ions outside the cell and import of potassium ions into the cell, and which is this one. This is the guy, the sodium potassium pump. It pumps 3 sodium ions outside the cell in return for 2 potassium ions into the cell, and uses ATP to maintain it. So this sodium potassium pump, what it essentially does is that maintains a higher concentration of sodium outside the cell and a low concentration of sodium inside the cell. And for potassium it is opposite. There is a higher concentration of potassium inside the cell and a low concentration of potassium outside the cell. So during action potential, what happens is that there is an initial stimulus, and this initial stimulus raises the membrane potential of the neuron to a threshold potential, which could be around minus 55 millivolts. Once this stage is reached, then there is opening up of voltage gated sodium channels. Voltage gated sodium channels are channels which allow the passage of sodium into the cell, but they only open in response to voltage increase. So there is an increase to minus 55. So this increase actually opens these sodium channels, and the sodium ions rush right through the channels and go into the cell, because there is a lot more of them outside as compared to inside, and this increases the entire membrane potential and it shoots up. This increase towards positive of the membrane potential is known as depolarization. Next, once a peak of depolarization, which is around plus 40 or plus 50 millivolt, is reached, a slow moving potassium channel is opened. And this potassium channel, as you would know, that there is more potassium inside than outside, pushes the potassium out. And since positive ion is going out, the entire membrane potential goes towards negative. And around this time, the sodium channels are also closing up again. So this entire peak of membrane potential drops down and down to lower levels. And this lowering of the membrane potential towards negative is called repolarization. And this continues till the threshold voltage is crossed, and it continues even a bit further than the initial condition, because potassium channels are, as they are slow to open, they are also slow to close. And this particular part is known as hyperpolarization, in which the membrane potential drops even below the initial level, and then it mellows out to the initial condition and everything goes back to normal. This entire process is known as action potential, in which the membrane potential of the neuron jumps to a high level and then drops down. And this is how neurons send signals via them. This is the electric current that we see when we say that neurons help in transduction of the signal. So redistribution occurs. There is less charge inside the cell than outside, and this is known as depolarization. Less negative charge inside, so this depolarization. Any questions? Okay. Next, efferent neurons, also called motor neurons, are the nerve fibers responsible for carrying signals from the blank in order to initiate an action. The options are brain to peripheral nervous system, peripheral nervous system to the brain, both A and B, none of A and B. So motor neurons carry signals from which part of the nervous system to which part. So you can put your answers into the chat. Surajit says it is A. Efferent neurons carry signals from the brain to the peripheral nervous system. Lavanya also says A. Jagruti also agrees that it is A, brain to the peripheral nervous system. Divya also agrees that it is A. They carry signals from the brain to the peripheral nervous system. Okay, let us look at it. So this is a reflex arc that we are looking at. So as we have in the initial slide seen how exactly the stimulus travels in our body. First, a sensor which might be placed on the skin receives the information using a receptor. This signal goes via the afferent nerve to the spinal cord, where the information travels to the efferent nerve, which carries the signal to the muscle or the effector, which then contracts or does whatever effect requires to retain homeostasis. So the pathway is receptor, afferent nerve, central nervous system processing unit, could be spinal cord or the brain, then the efferent nerve, and finally the muscle. Thus, efferent neuron, also called motor neuron, are the nerve fibers responsible for carrying signals from the brain to the peripheral nervous system. Okay, moving on. What happens in Alzheimer's disease? The options are: the cell bodies become larger, the cell bodies start to die off first, processes become demyelinated but alive while cell bodies die off, or processes start to die off. What exactly is the pathophysiology, or how exactly Alzheimer's disease occurs? You can put whatever you think is the correct option into the chat. Achal says it is D, processes start to die off. Surajit and Jagruti also say it is D, processes start to die off. Anyone else? Okay, I have a video here as well, but I will skip this. So these videos are for you to, so we have already covered all these videos, as you would know, during the original classes when we had covered these questions. So I am not going through all the videos, so I would encourage you to go through them after you receive the PowerPoint on your end. So during Alzheimer's, so this occurs. So can anyone tell me in which part of the brain does Alzheimer's occur? Does anyone have any idea? Which part of the brain does Alzheimer's occur in? Hippocampal region, you're correct. Alzheimer's occurs in the hippocampus, which is the seat of the memory of in our body, where long-term memories are synthesized. So in the particular neurons in the hippocampus, normally there are chains of neurons from one end to the other called CA1, CA2, CA3, CA4. These chains of neurons are very critical for memory formation. What happens during so normally you see that one neuron, which is shown right here, has its axon synapsing on the dendrite of the next neuron, and this is how the entire process occurs. So neurotransmitters are released into the synaptic cleft, which are detected by the postsynaptic neuron, and action potential occurs in the postsynaptic neuron, which essentially dictates that the signal is transferred. But in case of Alzheimer's disease, what happens is that there is aggregation of various proteins, especially the tau protein, which aggregates and disrupts the various microtubular arrangements that are present in the long axons of these neurons. Once these microtubular arrangements are disrupted, the ends of the axon, so right here, do not receive any organelles or nutrients or new proteins being synthesized by the nuclei here, because synthesis of most of these stuff occurs near the nuclei, and these need to be transferred to the axonal ends or axonal terminals. Once the microtubular arrangement, which actually is the arrangement via which these stuff are transferred, is disrupted, then we no longer have any transfer to the axonal end, which disrupts the entire signal transduction process, because the neural transmitters are also produced in the soma or the nucleus. So the axon starts dying off. The processes of the entire neuron start dying off, and this finally causes the neural death. And you can actually see neurofibrillary tangles in the outside of the cell as well as inside of the cell, where they disrupt the entire cellular transport. So what happens during Alzheimer's disease? The processes start to die off. So the correct answer is D. Next, at the neuromuscular junction, the synapse is formed between the presynaptic blank and the postsynaptic blank. So it is found between the presynaptic muscle and postsynaptic motor neuron, the presynaptic muscle and the postsynaptic muscle, the presynaptic motor neuron and the postsynaptic motor neuron, the presynaptic motor neuron and the postsynaptic muscle. So you have four options, A, B, C, D. You can put whatever you think is the correct option. Lavanya says D. It is formed between the presynaptic motor neuron and the postsynaptic muscle. Surajit also agrees that it is D, presynaptic motor neuron and the postsynaptic muscle. Jagruti also says that it is D. Anyone else wants to try and answer this? Okay. So another video here. Okay, so I think I will also explain this in detail instead of going to a video. Okay. So here we have a muscle cell as shown right here. You can see the sarcomeres represented schematically here, and this is where the motor neuron innervates the muscle. So the motor neuron has formed a synapse with the muscle right here, and once action potential comes through the neuron, it releases its neurotransmitters into the synaptic cleft of the neuron and the muscle. The muscle has various channels which are present on the synaptic cleft, and these are ligand based channels. So they bind to the neurotransmitters, which is most of the time acetylcholine, and acetylcholine channels open and cations can go in. This increases or depolarizes the membrane of the muscle. This signal of depolarization travels deep into the muscle via the T tubules or the transverse tubules, which signals release of calcium ions from the sarcoplasmic reticulum into the muscle syncytium. And the release of the calcium ions, as you have seen previously, goes and binds to the troponin, removes the tropomyosin, allows interaction between the actin and the myosin, and power stroke occurs and the muscle contracts. So the entire pathway Action potential in the motor neuron, release of acetylcholine. Acetylcholine is detected by the ligands present in the muscle synapse. Cations go in causes a depolarization. This causes an action potential in the muscle, which signals to release calcium ions from the sarcoplasmic reticulum. Calcium ions release, go in deep into the muscle, where the sarcomere is stopped by the tropomyosin from interacting. Calcium binds to the troponin, removes the tropomyosin, and the actin myosin interact to cause the power stroke and contraction of the muscle. Is that all okay? Is that fine? Do you want me to repeat this? Okay, I guess not. So moving forward, at the neuromuscular junction, the synapse is formed between the presynaptic motor neuron and the postsynaptic muscle. So the correct option is option D. Moving forward, in the dark, the photoreceptor is depolarized due to influx of blank through open ion channels that are gated by blank. So the options are, they are depolarized due to influx of sodium and calcium, and which are gated by cGMP. Sodium and potassium gated by calcium. Potassium and cGMP gated by calcium. Potassium and calcium gated by sodium. So Jagrati answers that the photoreceptors are depolarized due to influx of sodium and calcium gated by cGMP. Surajit also agrees to the fact. Preeti also says photoreceptor is depolarized due to an influx of sodium and calcium through open ion channels that are gated by cGMP. Okay, so here this video is important, so I would like you to go through this. Let me know if this is audible.
Last video, we looked at how light...
Is this audible? I did not get that. Is this audible? Okay.
From the sun can enter the eye, and this is the eye, so it enters the eye, hits the back of the eye, and somehow through a set of steps it gets converted into neural impulse, and the impulse gets sent to the brain for you to make sense of the information. So we looked at something called the retina in the last video, and we talked about how the retina is made up of a bunch of different cells, and the two main cells are rods and cones. So we're going to look at just the rods in this video. So the rod, which is shaped like this, let's give him a little smiley face because he's happy. And what he does is that as soon as light is presented to him, he basically takes that light and then converts it into a neural impulse. So normally he is turned on. When there's no light, the rod is turned on. But when light is present, it actually turns him off. So with light, he's turned off. So how does this happen? So this occurs through this thing called the phototransduction cascade. So the phototransduction cascade is this set of steps that occurs at the molecular level that basically takes this rod and turns him off. And in turning him off, he's actually able to turn on a bunch of other cells that eventually lets the brain know, hey, there's light here. So it allows the brain to realize that there's light and allows it to comprehend what's going on and make sense of the world. So let's go ahead and examine this phototransduction cascade. So let's give ourselves a little bit of space here to just look at it in a little bit more detail. So let's look at just a part of the rod. So I'm going to redraw the rod over here, And let's just look at this part of the rod, just this very top little bit of the rod. And I'm going to draw it a lot bigger. I'm going to draw it a lot bigger so that we can really make sense of what's going on here. So inside the rod, which we just made much bigger, there are a bunch of these little discs. So they're really thin little discs, and they're just stacked on top of one another, and there are hundreds of them. And they're like this. They just stacked on top of one another, and they fill up the entire rod. Same thing in cones, but we're just going to look at the rods here. So inside of these little discs, there are a whole bunch of different proteins, all interspersed throughout the discs. So this protein that I'm drawing in red, let me just go ahead and draw it a lot bigger. So we're going to go ahead and blow this protein up. And it's basically this multimeric protein, so it consists of a bunch of different subunits. There are actually 7 subunits, so there are 5, 6, and 7. So there are all these subunits that make up this little protein right here. So this protein as a whole is called rhodopsin. And it's called rhodopsin because it's in a rod. If it were in a cone, it would be called conopsin, but it's basically the same protein. And sitting inside of this protein is a small molecule, and it sits just inside. Here, let's go ahead and just kind of just zoom in a little bit just so that we can make a little bit more sense of what we're looking at here. So let's go ahead and zoom in over here. So this little molecule is just sitting inside of rhodopsin, and it's kind of bent. You can see here how I drew it. It's just a little bit bent. So this little molecule is called retinal. And in this conformation, in this bent conformation, we call it 11-cis retinal. And so what happens is light comes in from the sun, goes in through the pupil, hits the retina, and then hits this little rod. Some of the light will actually hit this molecule. So it'll hit rhodopsin, and then it'll actually hit this molecule. So here's the light wave. It comes in, and it just hits the molecule right there. So an interesting thing happens when the light hits the molecule right at this region. And what it actually causes is it causes the retinal to actually change conformation. So it actually causes the light provides enough energy that the retinal goes from this bent conformation, and it causes it to look more like this. So I'll draw it over here. Draw a little carbon ring so it becomes straight. So it went from being bent to being straight. So the light does this, and basically it goes from being 11-cis retinal to all trans retinal. So when the retinal changes shape, it actually causes the rhodopsin molecule to also change shape. So the two are really closely linked. So when the retinal changes shape, the rhodopsin changes shape. So let's go ahead and pretend that it looks like this. So this region that I'm shading in right here is what the new rhodopsin looks like after the retinal has changed shape after the light hit it. So rhodopsin now changes shape, and that basically begins this big cascade of events. So what happens next is there's a molecule, and I'm going to draw that molecule right here in green, and it's made up of 3 different parts. So there's this alpha subunit, there's a beta subunit, and there's a gamma subunit. And this molecule as a whole is called transducin. So transducin. Oops. All right. Sorry here. So transducin. So transducin basically, as soon as retinal changes shape and causes rhodopsin to change shape, transducin breaks away from rhodopsin, and the alpha subunit actually comes over here to another part of the disc and binds to a protein called phosphodiesterase, which I'll just draw as this little box over here. So this little protein is called cyclic GMP phosphodiesterase, or PDE for short. So PDE, basically what it does, let me just go ahead and zoom out a little bit. What PDE does when it's activated is it takes cyclic GMP, which is floating all around the cell. It's a little molecule, it's tiny, and it basically takes the cyclic GMP and converts it into just regular GMP. So this basically reduces the concentration of cyclic GMP and increases the concentration of GMP. And the reason that this is important is because there's another channel over here. So there's a whole bunch of these sodium channels. So a whole bunch of sodium channels, and they're all over the cell. So they're just a whole bunch of them. And basically what they let the cell to do is they allow the cell to take in sodium from the outside. So let's just say there's a little sodium ion, and it allows it to come inside the cell. So in order for this sodium channel to be open, it actually needs cyclic GMP to be bound to it. So as long as cyclic GMP is bound, the channel is open. But as the concentration of cyclic GMP decreases because of the phosphodiesterase, it actually causes sodium channels to close. So now we have a closing of sodium channels, and now we basically have less sodium entering the cell. And as less sodium enters the cell, it actually causes the cell to hyperpolarize and turn off. So as the sodium channels close, it actually causes the rods to turn off. So turn off. So basically without light, the rods are on because these sodium channels are open. Sodium's flowing through and the rods are turned on. They can actually produce an action potential and activate the next cell and so on. But as soon as they're turned off, what happens is very interesting, because let's just look at this rod over here. So what happens is really interesting, because there's this other cell over here that is called the bipolar cell, and we'll just give it kind of a neutral face because it's bipolar. This cell, there are actually two different variants. So there are on-center and there are off-center bipolar cells. Off-center. So the on-center bipolar cells normally are being turned off when this rod cell is turned on. But as we mentioned, due to the phototransduction cascade, the rods turn off, which actually turns on the bipolar cell. So basically, on-center bipolar cells get turned on with light and get turned off when there's no light. So that's how they get their names. So when the bipolar cell gets turned on, it activates a retinal ganglion cell, which then sends an axon to the optic nerve and then into the brain. And so that process is known as the phototransduction cascade, and it basically allows your brain to recognize that there's light entering the eyeball.
Hello, am I audible? I guess I am. Okay, so that concludes about the phototransduction cascade. We'll just go through it in a bit. there's a dark current that always flows through the rhodopsin, the rod cells, and this is only inhibited in when there is light falling on them, and the process, as we just saw, is gated by CGMP and rhodopsin. Going into it in more detail. So when light falls on the rhodopsin molecule, the rhodopsin molecule changes into so the rhodopsin molecule throws out the transretinal, which then goes through an entire process to go back to rhodopsin. While this happens, the other part of rhodopsin, which is the protein-oriented part, goes and activates transducing, changing CGMP to GMP. Change of CGMP to GMP closes the ion channels and thus stops the dark current, and that is how we are able to visualize light, by turning off of the rod cells. So in the dark, the photoreceptor is depolarized due to an influx of sodium and calcium through open ion channels that are gated by CGMP. Is that okay? Anyone has any questions from the video? Okay, it's not. Moving on, the vena cava is a blank that carries blank. The options are: it is an artery carrying oxygenated blood to the heart, to the body, artery carrying deoxygenated blood from the heart to the body, vein oxygenated blood to the heart from the body, vein deoxygenated blood from the body to the heart. So you can put whatever you think is the correct option in the chat. Surajit says it is D. Vena cava is a vein that carries deoxygenated blood from the body to the heart. Jagrati also says that it is a D, a vein which carries deoxygenated blood from the body to the heart. Let us look at it. So this is a schematic showing the entire heart and how exactly it pumps. So we know that the heart is divided into 4 chambers: the right atrium, left atrium, right ventricle, left ventricle. All the blood from the body is collected into the inferior and the superior vena cavas and put into the right atrium. This is the first thing that occurs. The right atrium pumps it into the right ventricle, and the right ventricle pumps the blood right into the pulmonary artery, which goes through the lungs, and in the lungs the deoxygenated blood is oxygenated, and then this blood comes back to the heart via the pulmonary vein right into the left atrium. The left atrium pumps the blood back into the left ventricle, and the left ventricle pumps the blood throughout the body via the aorta. So the way this happens is that the deoxygenated blood is received in the right atrium, it goes to the right ventricle, then the left atrium and the left ventricle. So, but this does not occur in this particular fashion. The pumping of the heart does not occur in this particular fashion, even though blood flows in this fashion. While the right atrium is beating and pushing the blood into the right atrium, the left atrium beats and pushes blood into the left ventricle. So the actual beating of the heart is right atrium, left atrium, right ventricle, left ventricle, even though the blood flow is in the other direction. Right atrium to right ventricle, left atrium to left ventricle. But the pumping is right atrium followed by left atrium followed by right ventricle followed by left ventricle. So the vena cava is a vein which carries deoxygenated blood into the right atrium. So the vena cava is a vein which carries deoxygenated blood from the body to the heart. Any questions from this? If there are no questions, we will go to the next one. क्या मुझे 10 मिनट दे दो क्लास में, मुझे उसके बाद में आता. Okay. The next question is, Which of the following is not part of the respiratory system? The options are trachea, larynx, pharynx, and bronchi. We already have answers pooling in. Surajit says it is B, larynx. Jagruti and Preeti also says that larynx is not part of the respiratory system. Let us look at it. We have another video, but I will skip this one as well and go to the diagram of the lungs and explain from here. Okay, so the respiratory system, the gross anatomy of the respiratory system consists first of the nasal cavity, where the air enters into our body, and this particular nasal cavity, as you would know, consists of a lot of minute hairs as well as mucus particles, which prevent any particulate matter as well as viruses or harmful pathogenic bacteria from entering further into the respiratory system. This is followed by the pharynx, which is the point where the mouth and the nasal cavity join, followed by the larynx. Larynx is where we have our vocal cords, and finally they enter into the striated tube, which is the trachea, which is the windpipe that you can see here right at the bottom of the neck. This has various striations, as you can see, which can accommodate the increase in pressure when air is going through it. And finally, the trachea divides into two parts as it enters into the lungs, into the left and the right bronchi. And the bronchi diffuse into the lungs. Inside the lungs, the bronchi divide into minute, smaller vesicles, which are known as bronchioles. These bronchioles, as you can see right here, this is a bronchiole, which ends up in a sac-like organelle, sac-like structure, which is known as the alveoli. These are normally collapsed, but once the air enters, these expand and form a balloon-like structure. This is where exchange of gases occurs, and as you can see right here, these are very well diffused with the veins and arteries, and there are blood vessels going right very closely to these alveolar walls. And these alveolar walls allow diffusion of gases. As you would know, deoxygenated blood coming from the heart has a higher amount of carbon dioxide and oxygen as compared to the air mixture here in the alveoli. And thus, when exchange of gases occur, carbon dioxide goes out into the alveoli and oxygen gets in, and the entire blood gets oxygenated. And this is carried through the heart via the pulmonary vein. So this is how the entire alveolar gas exchange occurs. So we have in the respiratory system, we see that there is a trachea, which is the windpipe, the pharynx, which is where the nasal cavity and the cavity of a mouth meets, and the bronchi, which are the divisions of the trachea into the lungs. There is nothing as the larynx that is present in the respiratory system. Next, iodine deficiency leads to blank that primarily affects the blank. The options are: iodine deficiency causes goiter, which affects the adrenal glands; it causes scurvy, which affects the adrenal glands; goiter, which causes affects the thyroid glands; and scurvy, which affects the thyroid glands. So we have Jagruti, who says it is C. Iodine deficiency causes goiter, primarily due to affecting the thyroid gland. Divya and Surajit and Preeti all three agree that iodine deficiency leads to goiter that primarily affects the thyroid gland. Okay, then let us quickly look at it. So goiter is indeed a disease which occurs because of problems in the thyroid gland. In this particular disease, the thyroid gland swells up or enlarges and can be seen as a visible swelling in the neck. This causes a tightness in the throat, difficulty swallowing, coughing, hoarseness, and difficulty breathing. So a healthy thyroid is something like this. A goiter thyroid expands like this. The reason behind goiter is a decrease in iodine or iodine deficiency in our diet. So the thyroid gland, as you would know, produces the hormone thyroxine. 4 forms. And thyroxine is thyroxine, like thyroxine has very many iodine moieties, as you can see right here, 4 in T4, which actually causes it to do its function. So without the iodine, the thyroxine cannot be made. So once there is a deficiency of iodine, the thyroid gland essentially goes into overdrive. It thinks that the thyroid gland is not able to produce because there are not enough thyroxine producing cells present in it. And thus it starts expanding to actually produce more thyroxine. But since there is no iodine present in the diet, you are still not able to produce thyroxine. And this causes a vicious feedback loop, which causes the enlargement of the entire thyroid gland and finally causes goiter. So, coming back to the question, iodine deficiency leads to goiter that primarily affects the thyroid gland. Yeah. Any questions from this? Okay, so that is all I had for you today, and I guess that brings us a close to all of our PMRF-NPTEL sessions of this particular course. So I have tried to cover most of the important concepts in this particular PowerPoint from all the weeks that we have covered till date. But obviously there are other things that you should already know about this to better understand the entire concept behind the course, animal physiology, and it will help you throughout towards your final exam as well. So I would very much encourage you that you go through all the PowerPoints that we have gone through previously. All of them are uploaded to a Google Drive whose link is available both on the YouTube channel as well as the NPTEL videos channel, where exactly you see the NPTEL videos. It is available there as well, so you can go through it at your leisure to understand everything. And if you have any questions, then you can reach out to me via the YouTube comments or the NPTEL forum, yeah, and we can discuss it whenever you want. So does anyone have any questions from this? Anything throughout the course that you wanted to ask? Okay, if no one has any questions, then I guess I will bid you thank you for joining. Sir, is there negative marking for the final exam? I do not think so. There are any negative markings for the final exam. The entire format will be very similar to how your assignment questions were. So you will have MCQ question that you will answer, and it will be very similar to what you have been answering throughout the course. Okay, so thank you everyone for joining me for all these 12-13 weeks that we have been doing this PMRF-NPTEL sessions, and best of luck for your final exam and your future endeavors. So thank you everyone for joining.